Since the members of no species, even California redwoods or giant Galapagos tortoises, live forever, all species have to have members that successfully reproduce in order for the species to continue to survive. The process of reproduction requires that offspring obtain the genetic instructions or the blueprints of life contained in strands of DNA from their parent or parents. The two basic types of reproduction, asexual reproduction and sexual reproduction, differ in how they provide offspring with the genetic instructions that will define their traits and characteristics. During asexual reproduction, a means of reproduction often used by single-celled organisms, such as paramecium and amoebas, and certain multicellular organisms, such as hydra, a single parent passes exact copies of their genetic material on to the next generation through the process of mitosis. The result is offspring genetically identical to the parent. During sexual reproduction, the genetic material provided from two parents in the form of sexual gametes formed by the process of meiosis, which we'll look at shortly, join. The cell, created by the union of the two gametes, is called a zygote. The newly formed zygote has a genetic makeup that varies somewhat from that of either parent. The individual strands of DNA that carry the blueprints of life are called chromosomes. In eukaryotic organisms, or organisms made up of cells that have nuclei, chromosomes are found within the nucleus of each cell. Every living species has a characteristic number of chromosomes. For example, we humans have 46 chromosomes in the nuclei of all our cells except our sex cells, which have only 23. Chromosomes are made up of sections of DNA called genes. Each chromosome may have hundreds or thousands of genes. Most genes program cells to synthesize specific enzymes or other proteins. It is the cumulative action of these proteins that produces an organism's inherited traits. If we were to look at the chromosomes in the nucleus of a eukaryotic organism, we would notice that they could be distinguished from one another by their length, banding pattern, and the location of their centromere. In the cells of most organisms, we would also note that, except in their sex cells, two matching copies of each type of chromosome exist. These matching pairs of chromosomes are called homologs. One chromosome in each pair of homologs is inherited from the individual's male parent, and the other from their female parent during sexual reproduction. The two chromosomes in each homolog carry genes for the same inherited characteristics, and the gene or genes for each trait, such as eye color, are situated at identical locations on each chromosome in a homologous pair. Cells that have two matching sets of chromosomes are referred to as diploid cells. The bodies of most eukaryotic organisms, such as humans, are made up of diploid cells. When a single-cell diploid organism reproduces asexually via mitosis and cellular division, the offspring is assured of having diploid cells, because the process of mitosis and cellular division produces cells genetically identical to the parent and each other. Thus, a diploid cell reproducing through mitosis will always produce two diploid cells identical to the parent. But what if multicellular diploid organisms produced sex gametes, such as eggs and sperm, through mitosis, just as they produce the rest of their cells? If two such gametes joined, the zygote formed would have 4 copies of each homologous chromosome. The next generation of zygotes would have 8 copies, the next 16, and so on, each succeeding generation having double the number of chromosomes of the preceding one, until finally the number of chromosomes in the cells of each organism would become virtually infinite. Thus, in order for diploid organisms to reproduce sexually, they must produce sex gametes that have just a single copy of each homologous chromosome. so that the zygotes they produce are merely diploid like the parents. Cells like sex gametes, which have a single copy of each homologous chromosome in their nucleus, are referred to as haploid. The special form of cellular division by which organisms create haploid sex gametes is called meiosis. Meiosis not only forms sex gametes with one set of chromosomes, as we'll see later, it also moves genetic material around in ways that increase the genetic diversity within a species population. The competitive advantages arising from genetic diversity are probably the driving force behind the evolution of sexual reproduction in living organisms. While asexual reproduction, which often occurs in aspens and a variety of other species, such as Hydra, has a number of advantages over sexual reproduction, not the least of which are that individuals don't have to spend energy producing sex gametes or competing with sexual rivals, asexual reproduction has the major disadvantage that it doesn't create genetic diversity within the species as a whole. Lack of genetic diversity may not be that negative. In fact, it may be an advantage if the parent organism is well adapted to the environment. But what if the environment changes? For example, the climate becomes drier or a new predator enters the environment. If all the parents and offspring are genetically identical, the entire species may be destroyed as a result of one or two changes in the environment. However, if the members of a species vary somewhat genetically, and thus in some of their physical characteristics, it is much easier to imagine that at least some of the members of the species will have characteristics that will enable them to escape succumbing to a change in environment, such as less rain or the arrival of a new predator. This probably explains why even aspens occasionally reproduce sexually. Meiosis and sexual reproduction are means by which species create genetic diversity within their population. The process of producing gametes via meiosis takes place in the reproductive organs of an organism. In humans and other mammals, male gametes or sperm cells are produced in the testes of males, while female gametes or eggs are produced in the ovaries of females. To illustrate the process of meiosis, let's look at an imaginary male diploid animal with 3 homologous pairs of chromosomes. The process of meiosis begins in the testes of our imaginary organism, with diploid cells that are genetically identical to those in the rest of the body. Of the 2 chromosomes forming each of the 3 homologous pairs in the nucleus of these diploid cells, one came from the organism's male or paternal parent, and the other from their female or maternal parent. Before meiosis begins, both the paternal and maternal chromosomes that make up each homologous pair duplicate themselves. These copies remain attached to each other at their centromeres. Each copy of the original chromosome is called a chromatid. Even though each chromatid will eventually function as an individual chromosome, as long as the 2 chromatids are attached to one another, we will refer to them as a single chromosome. It's not until they separate later that we will refer to each as an independent chromosome. Meiosis is divided into 2 parts, meiosis I and meiosis II. Both meiosis I and II are divided into 4 phases: prophase, metaphase, anaphase, and telophase. During the first phase of meiosis I, prophase I, chromosomes begin to condense and the maternal and paternal chromosomes that form each homologous pair line up next to one another in a process called synapsis. Each complex of 4 chromatids produced by a homologous pair of chromosomes lining up next to one another is called a tetrad. Networks of protein strands join the homologs in each tetrad together, shifting their positions relative to one another until the corresponding genes on the homologs line up exactly with one another. After the genes of the homologs are lined up, special enzymes exchange corresponding sections of genes between the 2 homologs. In the course of exchanging these sections, the paternal and maternal chromosomes come to cross over one another at points along their length, called chiasmata. Besides reflecting where chromosomes have exchanged sections of genes, chiasmata also hold homologous pairs together until they are later separated. The exchanging of sections of genes between homologs is appropriately enough called crossing over. Crossing over, by creating chromosomes with unique combinations of genes, is one of the events that occur during meiosis that help increase the diversity of individuals within a species. While homologs are lining up and crossing over in the nucleus, outside in the cell cytoplasm, other processes critical to meiosis are also taking place. All eukaryotic cells have a structure called a microtubule organizing center, or centrosome, near the nucleus that synthesizes the protein tubulin. Tubulin is used to create long, thin filaments called microtubules. A network of these microtubules radiates out from the centrosome, helping to form and maintain the cell's shape. During meiosis, the network of microtubules radiating out from the centrosome disappears, and the centrosome divides into 2 new daughter centrosomes. The daughter centrosomes then move to opposite poles of the cell, each forming a network of microtubules that interconnect with the microtubules radiating from the other to form a structure called the spindle. The spindle consists of polar microtubules that radiate from either centrosome and overlap at the equator, and kinetochore microtubules, which will eventually attach to the chromosomes at their kinetochores, which are located near the centromere where the 2 sister chromatids are tied together. Once the centrosomes are at opposite ends of the cell and the spindle is completely formed, the nuclear envelope disintegrates, allowing the kinetochore microtubules to break in and attach to the chromosomes. Due to the lining up of chromosomes crossing over, and formation of the spindle, prophase 1 is by far the longest stage of meiosis, often accounting for over 90% of the time spent by cells in meiosis. The next phase of meiosis 1 is metaphase 1, during which the kinetochore microtubules move the homologous pairs of chromosomes to the equator of the cell, or what biologists refer to as the metaphase plate, where the 2 chromosomes in each tetrad lie directly across from one another. The chromosomes in each homologous pair are now positioned to be drawn to opposite poles. Which chromosome in a homologous pair is positioned to be drawn to a given pole of the spindle is completely random and totally independent of the positioning of any other homologous pair. This has a number of important ramifications, not the least of which is that if the genes for any 2 traits are on different chromosomes, those 2 traits will be inherited totally independently of one another. This results in what geneticists refer to as an independent assortment of genes. The independent assortment of homologous pairs means that species with a substantial number of chromosomes can create gametes with huge numbers of genetic combinations. For example, each human with their 23 homologous pairs of chromosomes can, just based on the random assortment of homologous pairs, produce 2 to the 23rd power, or approximately 8 million genetically different gametes. When we consider that human sexual reproduction occurs between 2 individuals, each capable of producing 8 million different gametes based on random assortment alone, we realize that the chances of any two children produced by a couple being genetically identical are over 1 in 64 trillion, except for the case of identical twins, which develop from the same sperm and egg. When we add variations due to crossover, which occur at about the rate of 3 per homologous pair in humans, the odds of any two children produced by a couple being identical become astronomical. Thus, any individual is extremely unique genetically, even within their own family, let alone the entire world. During anaphase 1, the randomly assorted chromosomes are separated and segregated to different ends of the cell as the chiasmata holding the homologous pairs of chromosomes together let go, and the homologs start their journey down the kinetochore microtubules to opposite poles of the spindle, where they eventually form 2 randomly assorted clusters of chromosomes at each end of the cell. Meiosis 1 concludes with telophase 1, during which the chromosomes conclude their journey to one end of the spindle or the other, and the spindle breaks down. What happens next depends on the species of organism. In most species, nuclear envelopes form around the chromosomes' clusters, while the chromosomes relax and decondense and the cell divides. As prophase 2 begins, a new spindle starts to form in each of the 2 new daughter cells, the chromosomes recondense, and the nuclear envelope dissolves. After the spindles are completely formed, the sister chromatids are grabbed by the kinetochore microtubules extending from opposite poles of the spindle. Metaphase 2 is marked by the chromosomes in each daughter cell being lined up at the cell's equator or metaphase plate. Note that unlike metaphase 1, where homologous pairs of chromosomes line up along the metaphase plate, in metaphase 2 it is individual chromosomes made up of 2 sister chromatids that line up at the metaphase plate. As anaphase 2 begins, the centromere that holds each of the sister chromatids together splits in 2, and the 2 sister chromatids separate into 2 individual chromosomes. Each of these new chromosomes works its way down its respective kinetochore microtubule to the pole of the spindle closest to it. Telophase 2 completes meiosis. The spindle in each daughter cell disassembles, and each divides to form 2 more daughter cells. In each of these 4 daughter cells, the chromosomes go back into their extended state, while a nuclear envelope forms around each cluster of chromosomes, producing a new nucleus for each cell. The cells are once again in interphase and beginning to build the proteins critical to the cell's growth and development into a mature sex gamete, in the case of our imaginary male animal, sperm. The result of meiosis is 4 haploid cells that have only 1 set of chromosomes, unlike their diploid parent cell that was formed by mitosis and therefore had 2 sets of chromosomes. Also note that as a result of crossing over and the independent assortment of chromosomes that occurs in meiosis 1, all of these cells are different genetically from one another. As our imaginary animal is a male, the cells formed by meiosis will develop into sperm. If one of these sperm happens to unite with an egg, produced in a similar manner by a female of the species, a diploid zygote will be formed that is genetically different from either of the parent organisms. The zygote will develop into an adult organism as a result of cellular growth and division. In the process of cellular division as it occurs in the new developing organism, exact copies of the chromosomes that paired initially to form the zygote are passed on to each new cell as a result of mitosis. Thus, both mitosis and meiosis are critical biological processes for our imaginary species as they are for all species of living organisms. Mitosis enables organisms to grow and develop by ensuring that each new cell resulting from cellular division receives exact copies of the genes it needs to function. Meiosis is critical to living organisms in that it provides the means by which diploid organisms form the haploid, genetically diverse sex cells necessary for sexual reproduction and the production of the genetically diverse offspring critical to increasing a species' odds of survival. Genetic variation not only increases a species' odds of survival, but along with the mutations that arise in the genes of sex gametes, it is the driving force behind adaptive change and the evolution of species.